Super. So I think we should start. And today is very unusual lecture because today I cannot give you a solid background. I can share with you what I intrinsically do not have. But so what I can do for you, I can show you a lot of different quirks of molecular biology, of system biology. So just going home, you will be interested to just try this yourself and just so become interested, become impressed. So let's start. The question of today's lectures: do we understand life? And most of you have seen this beautiful picture of Mycoplasma genitalium synthesized by Karr team, and this is very worldwide. Maybe some of you have seen that. And also the rule of this lecture, please, I want to get you inside. Well, if you don't understand anything, please raise your hand, just ask immediately, because it's not me who makes these lectures beautiful, but you. So if you don't understand, please feel free to disturb and ask, because probably some of you are shy and they want to ask something, but they cannot. So please feel free here. So we start from this beautiful picture of Mycoplasma genitalium, which it's called Synthia now, and we say that it's first organism synthesized from scratch. We say that it's the first synthetic organism. So let me show you how this synthetic organism is, and let me guide you through a list of publications of Karr team. So that is one of the first ones, and this is from 2003. And what is interesting about this FAG 5X164, what is interested within, this FAG was the first genome completely sequenced. That was done by Sanger et al. And what is the whole idea of this publication? You know the sequence of the FAG. You want to synthesize it part by part, then ligate, and then you can transform an E. coli. FAG's DNA starts transcribed, and then FAG release, you can pull down your FAG, sequence it, and see how good you basically was. So what was interesting here, they had to resequence it in order to be good, because quality of your FAG strongly depends on how good you was in sequencing. And also they were so good that they observed one lethal error per 500 base pairs. So that's the mechanism how they did it. So first design oligo, synthesize oligo, then on gel purify oligo, ligate, ligate again, purify, and then transform E. coli, then take one plaque of E. coli, put a virus, and then cut the DNA. And what is very interesting, they provided a technology to do this just in 2 weeks, very fast. So very fast quality assessment. And also, when they synthesized this FAG, they just from the beginning, they knew that in the end of the day, the aim is to synthesize a whole artificial chromosome for a small bacteria. So even in this case, they already discussed an ability in future to make this synthetic organism. So go next. We have Mycoplasma genitalium, smallest organism on Earth. And what is interesting about it, it has about around 500 of protein coding genes. It predicted to have 30% of genes are non-essential, and genome is very small. It's like 600 cases, very small. And so the idea is this is the whole metabolome. This is very simple in terms of biology, believe me, and you obviously don't need all of that proteins to just keep this very simple. And so this is a whole genome. They sequenced it, they knew it, and the idea is there is a transposon containing plasmid. with ITR transposon. You took this plasmid, then plasmid keeps an ability of organism to grow on one of, everyone is welcome. So this transposon makes an ability to grow on some media with specific antibiotic, so we can select transformed cell very easily just by growing on the media with this antibiotic. Then we can run transposon specific PCR. We have primer for transposon, we run PCR, we have a whole transcript. And then we can sequence PCR, and by sequencing PCR product, we know where we were, we know what gene was disrupted. And what they say is that they created Mycoplasma laboratorium, they reduced the number of essential genes. We have 482, they reduced this number to 382, and 100 of genes appear to be non-essential. How they knew it? Because when you do this lot of times, you just get on the plateau, you see that just making more and more of your experiment, you do not get more of the genes non-essential. But this is not existing organism, in fact, because we disrupt genes, yes, but we don't know how about this, how about additive of a factor of these mutations. For example, we can have two pathways. It can go, metabolomic can go right, can go left. You disturb either of them, right or left, your organism is alive. If you disturb both of them, your organism is dead, because there is nothing to compensate. But here they have nothing about non-additive mutations. They just, what they get in the end of the day, they get a list of genes which appear to be non-essential and nothing more. You don't know which of these two non-essential genes you can combine in one organism. So let's go into the next publication, and these methods become extremely powerful. Now you can transform a whole bacteria with DNA from another bacteria. So we have two very small organisms. One of these organisms is Mycoplasma capricolum, one is Mycoplasma mycoides. And we take Mycoplasma mycoides, purify its DNA whole chromosome, purify it from all of the proteins, and use it so as to transform Mycoplasma mycoides or Mycoplasma capricolum. These two organisms are very similar, and in terms of protein coding genes, 85% of protein coding genes of Mycoplasma mycoides correspond to Mycoplasma capricolum. So with this similarity, there is a high probability that you will be able to convert one organism into another. So that's the end of a whole thing. This is advanced glycol mediated transformation, and what you can see from this picture, this is Mycoplasma mycoides to two-dimensional gene electrophoresis, and this is Mycoplasma capricolum, and you can see nothing similar between two of that, but almost everything is similar for Mycoplasma mycoides and for our transplantant clone. So what we did, we successfully converted one species into another. So go next. This is extremely powerful. And each year we become better and better in sequencing. Each year we become better and better in synthesizing genomes. So now they see that they have an ability to synthesize a whole genome of Mycoplasma genitalium with low error rate. And idea is you synthesize these very short fragments, then you can ligate them in E. coli, and then there is a YAG artificial chromosome, and with this help of YAG artificial chromosome, you can use a natural recombination mechanism to, in the end of the day, assembly a whole Mycoplasma genitalium genome completely in yeast. I don't know what happened to this yeast. It maybe wasn't too happy to carry a whole Mycoplasma genome chromosome, but it was alive, And now we can purify whole chromosome from the east and go here and sequence it. So what they also did, they include small, tiny specific sequences. They named it watermarks. And by sequencing Mycoplasma genitalium whole genome, you can see that this chromosome is artificial. This is done by scientists. So again, they synthesize it, and when you have both of these two things, you can first make chromosome very beautiful with low mistake rate, and you can create a whole bacterial cell. Why do not combine this together? Why do not make chromosome one? First make chromosome and then use this chromosome to transform. And there is still a scientific sense, a lot of it, because when you purify, how you can prove that we purified almost 100% of the Mycoplasma mycoides genome. So idea is synthesize from scratch, similar mechanism, synthesize oligo, ligate oligo in put in E. coli, assemble yeast, and in the end of the day get a whole chromosome in yeast, purify from yeast, and then transform it Mycoplasma capricolium to make Mycoplasma mycoides. And initially, what is interesting, initially transformation failed, and because they have a common restriction methylation system, and also this whole genome, this 1,000 of 1 megabase was completely non-working because a single mutation and one gene essential for survival, a DNA A. So they uncovered this mutation, corrected it, and yes, they get a working Mycoplasma mycoides completely from scratch. So, and this is beautiful picture. They also wrote their names in A, coded it, and Mycoplasma mycoides codes also for leg Z. Leg Z makes this beautiful color, blue, and new organism now is called Synthia. So let's summarize what they did. They did first bacteriophage synthesis. Second, they did gene deletion studies and studied more than 5,000 deletions. And what is this is very common now because quite frequently for small organism we transform small organism with our plasmid containing transposon, and after that we have a list of non-essential genes. So it's frequently done, common technique. So next, they did an advanced polyglycol mediated transformation. I converted one species to another. They did whole genome high quality synthesis, and then they combined this one and created from a Mycoplasma capricolium. And here they are very predictable, and what they do next, want to synthesize Mycoplasma laboratorium. They want to combine together this part, gene deletion studies, whole genome high quality synthesis, and advanced transformation together with this. And so what you can get from here, you can get a minimal organism synthesized completely from scratch. It's not been synthesized yet, but they're going to do it. And this is for reason, for example, for optimizing fuel production, because the less genes not necessary for survival you have, the more productive organism probably you are, and so on. So they want to do it. But the question of today's lecture is, have they really created a life? The answer can be yes and can be no. This is a very philosophical question. Maybe they successfully created a life, they successfully booted it from computer. We don't know if they really created it. So Greg Venter says, this is the first organism which father is a computer. But to introduce like some comments about that, when we have a programmer, and programmer writes a code on Python, for example, Python is, programmer is a father of this program, yes, right? We do not say that our flash card on our computer is a father of this program. So probably they have to say that successfully booted already pre-existing life from computer. And I really like these words. This is the case when software writes its own hardware. And this is really right, because we have this software, this DNA, and DNA, a software, builds a whole organism around itself, its hardware. And that's how it works. But we want to know, do we understand life? And creation of new life, in this case, I don't think it provided much of understanding. Because what we know, we can convert one species to another. We know that essential parts, but we want to be clear what we understand about life. So, again, answer for this question, do we understand life? Possible can be, we know all these molecular processes very simple. We know how they organize phosphorylation. We know metabolomics. We know how transcription starts, how translation starts. All of that are in books. So maybe we understand life, but let me introduce some beauty here. Probably most of you are familiar with this concept by Jacob and Monod. Then in bacteria we have an operon. And what you have to know about this, we have RNA polymerase. RNA polymerase starts and make a polycystron transcript, and polycystron transcript is then binds, and from the polycystron transcript makes, there is four proteins, and it makes 4 proteins. So very simple. In the case of regulation, we have its majority dependent on this sigma factor, and by changing this sigma factor, it binds to different promoters. And also there is 2 kinds of termination, rho-dependent and rho-independent termination. So that's basically what you have to know. But next, molecular biology moves forward, and then we see that there are some oddities. Some oddities like there are RNA polymerase associated protein which strongly affect transcription. For example, there is one transcription start site, second transcription start site, separated by 5 base pairs. It starts from first transcription starting site, it makes long transcript. It starts from second transcription started, it makes very short transcript. So in this case, we for sure know that recognition of the termination signals strongly depend on what proteins were in the beginning with this RNA polymerase. Next, we know some oddities like internal promoters within operons. Very frequently we have, for example, promoter in the middle of operons. We have small RNAs, which add a lot of complexity. We have riboswitches, which also probably it's, I don't know, maybe the ancient mechanism, quite possible, it's the most ancient mechanism of gene regulation. We have non-canonical ribosome binding motifs. We have read-through terminators. But system biology moves, possibly somewhere even replaces molecular biology, and now we started to quantify all of that. And then we see that operon concept is, what is written here, it's under revision, but frankly speaking, now operon concept is dead. So let's start. Promoter. A lot of genes are transcribed, it's very frequent, a lot of genes are transcribed more than from one promoter. A lot of times we see this read-through termination signal, and a lot of times we see that regulatory information encoding genes can affect transcription and quantity of mRNA. For example, you have similar promoters. You take your promoter, you put like a list of genes, and you can see that by varying genes, you have different quantities of mRNA. So information in the coding part of the genes by itself strongly affect transcription. So next, small RNAs, also they add a lot of complexity. And next, this is very new method, ultrasequencing, high throughput, very sensitive to small RNA detection. And what we see now that we have in a 20% of the genes encode for a small RNA. And this is like in human, in plants, in yeast, and everywhere. So These bacteria are quite complex, but let's see something even more beautiful. We thought that mRNA in bacteria are degraded very fast. But for example, now we know that a lot of enzymes affect the mRNA stability. For example, we have this enzyme RPPH, which takes 5' phosphate, and after that mRNA degraded very, very quickly. And also RNA stability is affected by RNA binding proteins. And contrary to common view, in bacteria we have a polyadenylation. And there is two systems of polyadenylation in bacteria. I have no clue why there is still in notebooks from the university in molecular biology. But for example, in Bacillus subtilis, 20% of total mRNA is polyadenylated. And in E. coli, we have 70% of mRNA which needs raw factor for termination. 72% of this mRNA are polyadenylated in the end. But in the case of bacteria, polyadenylation, instead of giving longer life to our transcript, it promotes a very, very fast degradation. And finally, we found a protein splicing, but in endosymbionts. So a lot of complexity, but that's not all. What about Shiner-Dalgarno sequence? What was said before, in order to get this high quality translation, you need a very good Shiner-Dalgarno sequencing. But what we are getting more and more, that quite often we do not need at all this Shiner-Dalgarno sequencing. There are non-canonical ribosome binding sites. Also, there are different kinds of ribosome which recognize a different ribosome binding site. And also, polycystron mRNA are polar. That means if you have mRNA, that doesn't mean that you have the same quantity of the protein translated from each protein coding part of the mRNA. So next, genome tree reorganization, very complicated, strongly affects transcription, but it's not really well studied now how. And mRNA localization, this is very beautiful. So what we have to know here, and when mRNA is synthesized, it doesn't go anywhere. It diffuses 2 orders of magnitude slower than we would expect by simple stochasticity. It still stays in the same place. And this is a nice way, for example, we have protein complexes, and we can use a DNA, nucleoid, whole structure, so to assemble and bring in close proximity proteins which can, in the end of the day, assemble and make a protein complex. So this is a very efficient way to just put these proteins from same complex in a close proximity. And now the term of transcriptional factories is proposed.
How it realizes?
What?
How it realizes in cells?
Can you please ask
how
longer?
It works? I don't understand exactly how this mechanism works.
Why mRNA
Why
don't...
it don't diffuse from...
I still haven't seen the mechanism why it happens. So probably this is a question of molecular biology, why there is no diffusion. Probably there is a protein which binds, for example, recognizes a DNA and simultaneously recognizes mRNA. So
there are special sequences in the structure of RNA. Maybe
we do not know.
terminal.
Still, we don't know why this do not diffuses.
In cells, maybe some like proteins. For example, they have N-terminal.
Of course, and
Maybe the same way is for mRNA.
probably it will be a big hit parade if we find. But now we didn't find any protein targeting in bacteria, and still we didn't find mRNA targeting. And yes, maybe it can look like a protein targeting, But now at least I haven't seen an article which describes why this mRNA do not diffuses freely from the DNA. Probably some proteins with binding affinity or something like that. Probably also it could bind to cytoskeleton unspecifically, because in bacteria we have this whole unspecific cytoskeleton. For example, you have just protein, here it binds on one side. For example, intermediate filaments or FTZ, analog of tubulin in bacteria. And so it can keep mRNA on the same place. I haven't seen exactly the molecular mechanism, and I am really interested to see it. So let's go next. That comes from EMBL, Luis Serrano and Borg. They had an idea. What if we put everything against one organism? So we have these beautiful methods. We have protein quantification, mass spec. This is what they used, but not only one method to define protein complexes, there are plenty of them. But what they used here, it's just type affinity purification. You have an covered with immunoglobulin, you pull your complex once, then you cut, pull it second, and then this complex goes to mass spec to identify which proteins you had. And they also did a structural reconstruction by electron microscopy. You have this electron microscopy, you can predict the structure of the proteins, and then you can assemble a whole cell in 3D with 3D structure. And by the way, this method now is extremely powerful. I have seen the same thing for yeast. And what the scientists did for yeast, they just cut yeast into parts, then it goes into electron microscopy, and in the end of then you see complexes, and in the end of the day scientists were able to reconstruct 3/4 of protein complexes into yeast simply by EM. So also we have these beautiful methods like NMR, GCM, LCMS for metabolomics fluxes. We have chip, a lot of the DNA pull down, and all these methods to find protein binding sites on DNA. We have mRNA expression, microarray, Thylene array. So Thylene array, I think all of you are familiar with microarray, yes? And about Thylene array, it's basically the same, but that's for whole genome. So the whole genome cuts for parts. Also you have to put here a mutants, and then you can integrate all of this data about one very simplest organism. Not simplest, but very simple. And you can integrate this data together in computer. So what you want to get? You want to predict each molecule, whole cell, and understand 100%. Ideally, you would press button and new bacteria would divide in reproducing itself in computer. So let's start. Just to get you idea what is this Mycoplasma pneumonia, and it has like 700 of open reading frames. It has 44 RNAs. It had essentiality transposome analysis done. In the case of transcription, it has just 10 classical transcription factors and also non-classical DNA binding proteins, full repertoire for chromatin compaction and remodeling. And in case of signalin, it has 2 serine-threonine protein kinases and 1 phosphatase. So what can be simpler, this way of regulation? And almost full chemical signal and repertoire is just these 3 chemicals, APPPA, PPGPP, and CPGMP. So again, what can be simpler than this organism? So we start to reconstruct metabolic network, and first we want to define a minimal media. We go to 1,300 different media to define which components are essential, which are not, so as to exclude contamination from the media. And in the end of the day, they want to get this curated metabolic log. So now programs are extremely powerful. You sequence new organism, then you put this into KEGG, and what KEGG gives to you, you have like a whole metabolome just from genome. But the problem is the quality of is this really low, and what Luis personally says, and everything was disrupted, nothing connected, and they had to manually go through to correct reactions, so see what goes where. And for example, you know this standard lipid biosynthesis pathway, yes? You know all these steps, and for some reason one step is missing, and Keck doesn't identify it. And then you can go through, do BLAST, for example, look, maybe you have a whole pathway in this organism, but maybe you can, by different method, identify this enzyme, missing and identify missing reaction. So, yeah.
By comparison to
Oh, this is not.
organism, or how?
Sorry, this is not a program. This is like a completion of the program. So, just correct it with the term. And what you wanted to ask?
How this constructs the metabolomic maps? It's
How Keck works?
Utilize comparison with other
Yes. You have, for example, for E. coli, for yeast, you have these beautiful complexes, you have these proteins. So what it basically does is, in a very simplified view, you have your genome sequence, you can define proteins, this is simple in case of bacteria. You can define your protein, then you go BLAST, compare it to E. coli or to yeast, find something similar, and then you say, okay, that is that protein. It's more complicated. I am personally from bioinformatics, and what I can say to you, this is more complicated because sometimes there are different profiles, there are different constructions. Sometimes you can go into 3D structure and then compare enzymes. So there are a lot of methods which you can use to infer the function of enzyme. So what we get from molecular biology is that we have, for example, enzyme which catalyzes one reaction. It always adds phosphate, for example, to glucose. But what we have in reality, a lot of moonlighting, and depending on the quantity of metabolites, it can perform the same reaction, but chemical reaction is the same, but depending on the concentration on different compounds in the media, and can perform this with different compounds. So that wasn't the end of metabolomics map. So what they did, and they did one thing, what is called flux balance analysis. And you have the basics of flux balance analysis. You have stoichiometric coefficients, and you know all of these reactions. And you try, for example, optimize the gross mass production, and you can see which pathways are more essential for terms of energy or for terms of metabolites. And also this helps a lot to identify missed reactions. So they did flux balance, and in the end of the day, they published this beautiful article. What they had? We have complete metabolomic map. We have concentration dynamics. We have organism parameters in real time. We have essentiality analysis done. What we miss? We miss parameters for reaction. For example, we do not have a clue, for example, which is IC50 constant for this protein, which is the speed of conversion. And we can them from Brenda, but in case of Brenda, you will have that for E. coli. Also, about regulatory loops, they missed them because there's still no massive method to identify which chemical goes where and regulates what. And also some enzymes are missed. And in the end of the day, they published this beautiful article, impact of genome reduction on bacterial metabolism, and nothing they could do more. So the transcriptome. This is very complicated. You have microarrays, tiling arrays, deep sequencing. Deep sequencing is for the purpose of tiling arrays, because you have to know precisely your sequence in order to have these primes. You have DNA pull down, ChIP-seq, ASMRT. ASMRT, it's a special technique for those of you who are not familiar. this technique is to identify a methylation site in DNA. Also, what interesting they found, then enzymes are frequently transcriptional factors. Very beautiful way for regulation. For example, we have an enzyme, and if this enzyme can bind DNA, and normally this enzyme binds DNA in its catalytic site. That's what most often happens, because often the biggest pocket that's the pocket when you bind DNA, you both perform your catalytic function, but that doesn't really matter. So when there is a reaction, there is a compound in the media, what it does, it catalyzes a reaction. And if there is no reaction, so it sits on the DNA and works as a transcriptional factor. And this is a very frequent mechanism. Also, response without transcriptional factor, we knew that they have 10, but complexity of responses to different perturbations from the media is very big, and sometimes you even don't have an enzyme, and that's the very general problem of system biology. For most of networks in system biology, for protein interaction network, for transcription networks, quite often we don't have a clue which a response starts from. And possibilities here, for example, let me make it more clear. You have a Bacillus subtilis, you have a Mycoplasma pneumonia. In the case of Bacillus subtilis, you have a profile of responding for some kind of perturbation, for example, osmotic response. And the same proteins are expressed in the case of osmotic response, both in Mycoplasma pneumonia and in Bacillus subtilis. But in the case of Mycoplasma pneumonia, you simply don't have transcription factors. So what can contribute? Maybe other proteins can contribute, maybe non-coding RNA can contribute, maybe chromatin remodeling can contribute, methylation, specific DNA degradation, and so on. So also for people who like quirks, this is tiny RNA. Tiny RNA, some of you heard about that. So what tiny RNA does, it binds downstream from the promoter to the protein or non-protein coding RNA. They are 20 to 50 bases long, and still about tiny RNA, this is special kind of small RNA, and still for tiny RNA we have no clue what they do. We think that, for example, if we have RNA polymerase and we have this tiny RNA when we start transcription, and transcription go, tiny RNA bounds, and if there was a wrong recognition, the RNA polymerase had a very good chance not to reach the end of the gene. It's just abrupted from the beginning. Probably it's that, but still I haven't seen an article where someone proved that.
What is the origin of such RNAs? From what things they are processed?
For example, there are two possible origins. You can get them from the same gene but opposite direction, or they can be in a different gene. So two possible origins. Clear?
Yeah, but how we can replace the level of antisense transcription in it, on the gene? We need a lot of factors, and I say no, in Mycoplasma there is a very few proteins, you say to us.
Yes, there are very few proteins, but again, what you wanted, I cannot clearly understand the question.
How can we replace the transcription from antisense strand? We need to replace the level of transcription.
Of course, but
how?
There are a lot of possible mechanisms for that, and any way of regulation that is suitable for a protein coding gene or non-protein coding gene will be also suitable for a tiny RNA.
But
so you are interested how we get this RNA, how they are processed, so mechanism of how we cut them. Oh, clearly I don't know how probably they are done in the same way. Of course, probably they are done in the same way as all other smaller RNAs. So you make a longer transcript, then counted parts, and So you asked whether we have an enzyme to cut in parts the transcript.
Somewhat like this.
Something like that. Oh, I clearly didn't notice it. But probably yes. Probably we don't make this just short transcripts. And so when you do this mass spec, it's somewhat different from molecular biology, because you don't really care why you get that, because you just know that you have that, and only then you go into molecular mechanism. You know that you have a lot of tiny RNAs, but still you may not have a clue.
We only know that this mechanism is real, but we know how it works.
Yes.
Okay.
In this case, probably yes, but maybe for example, they are in plants, in yeast, and also in human, and probably for different clades we understand, but I didn't really look about how do we get them. So what about the transcriptome? What we have in the end of the day? We have analysis with different perturbations, in this case like 60 different perturbations from the media. We have accurate transcription data. We have a chip sequencing for most transcriptional factors, so we know where they bind. We have promoters, and what we miss? We miss who regulates transcriptional factors. We miss chromatin structure in 3D, and we miss how chromatin structure can affect the transcripts. So a lot of things remains to be done. Again, they published this beautiful paper, and nothing they could do more. And let's jump into proteomics. Just to simplify this view, their aim is by using different techniques. First, they want to quantify each single protein in the cell in different points of cell cycle. They want to remodel protein complexes, and they want to reconstruct a whole cell 3D. So what is interesting here about the proteomics, this is not really unusual. Just a few proteins time compromise as a large percentage of the mass. This is what we're waiting for, because we have a lot of this basic cellular machinery, and of course we will be waiting that they are really needed very frequently for example, polymerase, DNA polymerase, different pyruvate kinases, and so on. And also, you may already know that neither of the proteins performs its function alone. Most of the proteins can be found in complexes. And also, what is very interesting, ribosomal proteins can be found in complex with metabolic enzymes. This is not really surprising and very logical, because the majority of energy from the cell can go when the cell grows, up to 7% can go into protein synthesis. And of course, there is not very surprising that you have a crosstalk with a protein synthesis and metabolism. And what is also interesting, most of the cells have 0 copies of particular RNA in the cell. So that means in a whole population you have this. But for just this particular cell in this moment of time, you don't have any. Also, there is quite often what is very common view. For example, if mRNA goes up, the protein also goes up. So that's what we often think. But sometimes we have there is no correlation. Sometimes it's anti-correlation. mRNA goes up, protein goes down. So why this happens? A lot of different reasons. One of the reasons, for example, they have a very different half-life times. RNA is very fastly degraded. And also, mycoplasma pneumonia is under this uniform constant, like it seems that each organism is under that we have 100 till 200 grams of the protein per liter in living cell culture. So this is very uniform, because we find this in yeast, human, plant, and everywhere. So proteomics, electron microscopy tomography, we have complexes, dot complexes here, and we have this nice picture of whole cell in 3D, and we see where what is situated. Next, what is the proteomics? What we have? We have absolute copy number for proteins and ribosomes. We have protein half-life. We have absolute numbers. We have this beautiful picture of 3D cell. And also we are able to identify phosphoproteome. This is not really complicated. What we miss, there is still no mass method for accurately predicting RNA half-life. We still don't know why there is so frequently we find anti-correlation between level of mRNA and proteins. And also we miss effects of post-translational modification, but that can be done by mass methods. And finally, they published these beautiful papers. Not two, there are more than two of them, but I just pasted here, I thought most important. So nothing they could do more. So with all these mass methods, do we understand life? We are quite close. We quantify a lot of the proteins in the cell, but we thought we understand operon, and it remains to be very different. So a different group of Karr et al. Used the same idea, and this year in July or June, I cannot remember, that really not so important, and they predicted a host cell in computer, and protein by protein, reaction by reaction, you go and you can predict what happens. What is a little bit different, that they read a lot of publications and pasted the parameters for reactions, and now this organism facilitates the rate of biological discovery. So how we can optimize a whole cell? Here you can see that they measured this much of parameters, and how you can optimize a graph with that much of parameters, because you want this cell, you want to optimize what you have in computer to what you have in real life. For example, I want to have this slide. For example, we have a training data, yes, and we want to reproduce in computer what we have in experiment. So here the clue is to divide the whole cell into different models. For example, RNA decay can be a model. For this model you have this model parameters. RNA modification is model. Protein transcriptional regulation, macromolar complexation, all of these are models, and optimizing model by model, you can optimize each model independently, and then to construct a whole cell, you put these 28 models together and then optimize the whole cell, and you get this whole cell in computer. So that's how they looked. In quotes you can see how many genes are involved in each models, and how it goes. We just update time parameters, update concentrations, update several of our 1090 parameters, and go back, and so as to predict a whole cell. And what we have training data. For example, we have this multiplication time, we have how protein grows in the cell, how RNA grows in the cell, and what is interesting here. And for example, now you can predict data which you didn't have. For example, this is mRNA levels, and with this mRNA levels you can see this like a burst, yes? This is 0, and you have this mRNA degraded, again 0, mRNA degraded, 0, and so on. And this is the level of the protein, and look like the level of the protein is heated up. So you have these pulses, and by modulating the pulses frequency, you get the protein level goes up. So this is not solid, it's just pulses, and well, pulses frequencies modulation, you modulate the expression of the protein. So now it really moves the barriers of exploration. For example, now we can see a whole chromosome. We can see how it's explored by different factors. For example, we know that RNA polymerase during the cell cycle most often occupies an abundant transcript RNA, when RNA polymerase most often occupies the GRC. Or what about, we can measure the number of collisions during the cell cycle. So we know that during the cell cycle there is a 30,000 of approximately 30,000 on average collisions on the proteins on the chromosome. And now we can even say that each 1 second approximately one protein from the chromosome is displayed. So that what we couldn't get with our experimental methods. So we know now that 50% of the chromosome is explored. Part of exploration is because of the RNA polymerase. And just from the beginning, this thing also gives us a new hypothesis about cell cycle regulation. For example, initiation time, it can vary about 64%. Replication time, it vary around 40%. How overall cell cycle time can vary just by less than %. How that is possible? So, and there is non-transcriptional hypothesis here. This model gives us this hypothesis. So, if we start initiation of replication earlier, we have a fewer number of dNTP, and dNTP replication in energy consuming process in the case of dNTP. And if you start earlier, you will go over and wait until the dNTP level goes up. And if you start later, you just faster because there is no reason to wait for dNTP. So it corrects itself. Started earlier, replication longer. Started earlier, replication longer. Started later, replication shorter. So we have this very small variety. And again, how it moves our barriers. Now we have a whole picture of this energy, and we know how much energy in the different points of cell cycle, how much goes in translation, how much energy goes in, for example, tRNA isolation, how much goes in transcription, how much goes everywhere. We have these beautiful graphs for a period of cell cycle, and we know what we couldn't know before. Also, what is very good here, and again, they were able to simulate 128 wild-type cells. And what we often get if we run molecular dynamics, you know what is molecular dynamics? We just use force field, different forces, update time parameters, so as to run whole cell molecule by molecule. But it's a very energy consuming program because, for example, it's still very computationally demanding to fold one protein. And for example, there was a program called X where a lot of people joined their forces so as to fold one very small protein. And here they have a whole program written on MATLAB code. They didn't take into account this also folding. You don't look how each atom on the protein folds. You just say this protein is folded in this time. This complex is assembled in this time. So this is not really computationally demanding. But again, this is very powerful because you can predict a single gene deletions. And for example, here they tried to predict an effect of gene deletion on expression, and they did that for 20 single gene mutations, and in the case of 2/3 of the gene, they were in the order of magnitude. So their predicted level of expression from this model is consistent with what you have in real life. So now, do we understand life? And probably kind of. Now we understand 100% Mycoplasma genitalium, which is not very big, but you may ask, I want to understand human, and you want to understand human, okay, deal with this. This is human in terms of protein-protein interactions, and now we just starting to understand we are somewhere in the middle for the case of transcriptional networks, and we are just beginning this whole network understanding of the human, because still a lot of reactions are missing. We don't know, for example, there is weak physical binding, reaction is biological meaningful. So we just on the beginning of understanding of this very big and complicated thing. So that's why I really like science. Instead of giving answer on one question, you get a next question and even more. Do we understand transcriptional networks? Do we understand network? And so now we have new question, and I will make the topics of next lecture. Do we understand network? And on the next lecture we will learn properties and general principles of protein interaction network architecture. For example, these networks you cannot definitely see, but network consists of very densely connected evolutionary construct and modules. And in order to be fast, this network is a small word property. For example, you can get from this node to, any of these nodes very, very fast, not crossing too much number of these barriers. What it leads to, we have a protein hubs. A very few proteins are responsible for a majority of connection in this network. So this is hubs, and moreover we have different types of hubs. We have, for example, date hubs, party hubs, or sandwich interface, multiple interface hubs, and so on. This is very interesting. Also, we will learn something about quantification and interaction networks. For example, we can remodel a pathway, we can make a protein like a signalling in real time, and so on. What is very interesting here, for example, in network, there is a role for stochasticity. There are a lot of roles for stochasticity in protein interaction networks and in transcriptional networks. Also, we will learn about top-down and down-top synthetic biology approaches to understand network biology. So top-down approach, what will happen if we take our cell and rewire some pathway, and
induction, something like that.
What if we replace cyclin-dependent kinase 2 with something different? What if we remove some cyclins, or what if we rewire whole pathways? And there is also a reductionist approach, which I like even more. For example, we can construct what is called toy networks. For example, we can construct binary switch transcriptional. We can construct, you don't know what is circadian clocks, in how do we count time. And here we will talk about repressilator, how that was constructed, that a reductionist understanding. So everyone is welcome for the next lecture. Thank you for your attention. Thank you for questions, and I hope we have a useful discussion. Okay, any questions?
What was the cellular part of that models you talked about, of the synthetic organism? I think that they didn't construct the mycoplasma from water and lipids. I think, I think they had the cellular compartment for it, and what it was and how it affected.
Of course, that's the problem. Times, I don't know, maybe the problem of English. Sometimes we are missing things. So what they did, again, let's return to these publications. And they synthesized, what they did, they synthesized only DNA of well-known organism, Mycoplasma mycoides. And that's only what they did synthetic. Then they just transformed with this well-known DNA. They include the watermark and their names in this DNA, of course, to be more full. And then they transformed. And so the donor whole cell is still Mycoplasma capricolium. They just constructed a genome assembly. That's all, and then transformation. So they do not construct it from water, no, we are not on that level.
necessary genes.
Oh, okay.
Can you explain how works that transposon-associated construction?
Okay, you have plasmid. Plasmid contains transposon, and also we know that this transposon, when it comes cell, it makes it survivable under a certain condition. It makes it. It can survive with a different antibiotics. I cannot remember. Economicin may be different. I don't care. So we transform. When we transform, transposon goes out from this plasmid, goes into DNA. When it goes into DNA, it will not disrupt anything. Probably it will disrupt some genes. In the case of bacteria, most often it will disrupt genes because they don't have non-coding, too much non-coding. So it goes into genes and go into gene. If we have an insertion into gene, we assume that this gene is not working. Okay? Yeah. Then what we do? We have a specific primer. Specific primer recognized where our transposons are. What happens next with this specific primer? We go forward and make PCR. We see where our primer is. We see where our transposon is. And so by knowing where this transposon is, we can say, okay, if we have an insertion here, and still our cell is alive, yeah, probably this mutation is non-essential.
So the PCR goes between two transposons or inside the sequence of one?
Inside the sequence of one.